Alrighty, so welcome everyone to the second webinar of season 2 of the Hippocampal Subfields group webinar series. I'm really excited that today our featured topic is innovative approaches for subfield segmentations, and we have two phenomenal speakers here with us today, Dr. Sarah Genon and Dr. Ali Khan. And both of them have really innovative and cutting edge approaches, I think, to how we can examine the hippocampus. And as a group of people that are very interested in how we can answer questions that we haven't been able to previously, I'm really excited for us to be able to see what they have for us today and to be able to engage in some Q&A. So the way that it'll set up, if you haven't been here before, is that we will allow our speakers to give their talk and the presentation. You can ask questions in the chat box during if you'd like, but then after each of the talks finishes, we do reserve time for Q&A where we can have more of an interactive dialogue. And if there's time at the end, we can open it back up. So please feel free, if during a talk you want to post a question in the chat box, I'll be checking those throughout to make sure that the questions that you have are answered with the time we have available. But please make sure that you aren't unmuting or anything during the talk. We'll wait for the interactive part till the end. So with that, I'm going to hand it over to my wonderful co-host, Raban, for our introduction.
Thank you, Kelsey, and hi, everyone. It's my pleasure to introduce our first speaker for today, who is Professor Sarah Genon. Professor Genon did her PhD in psychological science at the Cyclotron Research Center at the University of Liège, Belgium. Then she did a postdoc in functional neuroanatomy at the Institute of Neuroscience and Medicine, Structural and Functional Organization of the Brain at the Research Center Jülich in Germany. And since 2019, Professor Genon leads the lab Cognitive Neuroinformatics and is particularly interested in understanding the principles of brain organization and how these relate to behavioral functions in humans. And today she will be presenting her work probing hippocampal organization in humans with parcellations and gradients. So, the floor is yours. You have 20 minutes for the talk.
Thank you very much for this nice introduction, and thank you very much for having invited me. I'm really happy to present our work today. So I will share my screen. Yep, you should be able to see it now. It should come in full screen in a second. Yeah, you see it full screen? Perfect. Okay, so our work aimed to study hippocampal organization in human with parcellation and gradients. So to first give you brief historical overview on brain parcellation. So brain parcellation is an umbrella term that actually covers a wide range of techniques that I use for studying brain organization. So it finds its roots in cartography. So the first cartography work was done based on histological materials with famous people like Brodmann. So what you see here is the Brodmann map that you may have recognized. And so the idea was to look under the microscope at the properties of the brain tissues to define different brain territories and then to manually draw a map. And then across the following centuries, the mapping has been automated, so still based on histology, but maps were automatically generated with computational approach. And this gave us cytoarchitecture mapping freely available, for example, in the U-brain. But a lot of progress came with the advent of neuroimaging scanners. So because with neuroimaging scanner, we could then look at the whole brain directly and have information about connectivity between regions. So we now can use parcellation approach applied to voxels or vertices to derive brain map with still automated approach. And in particular, we can use many different neuroimaging features that probe different neurobiological aspects to derive different, with a lot of computational power, to derive different brain maps that we can then compare or integrate together to have multimodal atlas. In that framework, very popular approach is connectivity-based parcellation. So the basic idea is that from connectivity features derived from neuroimaging data, there are different types of connectivity feature. I will come back to that later, but the idea is always the same. If we are interested in looking into the organization of a specific brain region or divided into subregions for some computational purpose, let's take here the hippocampus. The idea is we look at the voxel within this region. And if we take two different voxels, so for example, the red guy here and the green guy there, if we look at the connectivity profile across the brain, we would see that the red voxel is more connected to frontal region, while the green voxel may be more connected to posterior region of the brain. And we can derive a connectivity profile for all voxel within the hippocampus. And by doing so, we can cluster the voxel based on their connectivity profile. So for example, we can define here a group of red voxels, say, and a group of green voxels. And there are many different neuroimaging features that can be used with such parcellation approach. A popular technique is to use resting state functional connectivity. So I guess you have all heard about resting state functional connectivity. But another type of functional connectivity, which is less well known, is based on task-based fMRI. So the idea is that to apply a meta-analytic approach to thousands of activation experiments, this gives us a robust profile of co-activation for region of voxels across many different experiments. And this technique is known as meta-analytic connectivity modeling. So the connectivity that we get from there is not dependent on a specific cognitive paradigm, but is average across a range of different cognitive paradigms. And we have a large database in which we can run this approach, such as BrainMap and Neuroscience. So these are the most typical functional connectivity approaches that we can use for parcellation, but we can also use anatomical connectivity estimate from diffusion data, in particular with probabilistic diffusion tractography. However, I won't go into this approach today with regard to hippocampal parcellation, because the standard approach of probabilistic diffusion tractography, from our experience, doesn't work very well within the hippocampus. But there is a third technique that probe interaction between brain region, which is structural covariance. So which estimate comorphology between region. This is done, for example, using voxel-based morphometry and looking at covariance between voxel across a large sample of subject. So this is assumed to reflect coplasticity or co-atrophy between brain region. And we usually assume that comorphology between region reflects some kind of functional coupling between this region. But it may also reflect regions that share some microstructural properties. So we have different types of neuroimaging features that can be used for brain parcellation. And the general idea is that if we take a region of interest, the way it works, for example, here I pick up the dorsal premotor cortex just to illustrate the method. We use a connectivity matrix. So we take all voxel within this region that we call seed voxel, and all of the voxel in the brain, typically in gray matter. We compute a connectivity matrix of each seed voxel with all other whole brain voxel. Then we look at the similarity, the pairwise similarity or distance between the seed voxels. And we can apply a clustering algorithm, for example, k-means clustering, to define different clusters. So for example, here we have 5 different clusters among the seed voxel. And if we remap that to the brain, we see that how the dorsal premotor cortex can be subdivided into 5 subregions based on the connectivity profile of the voxel. Okay, so now we have a range of parcellation approach that can be used to better understand hippocampus organization. These methods goes from local microstructure to connectivity markers. We all, I think, are aware that from histological work in the hippocampus, different subfields have been defined, and they are usually spatially arranged along the medial lateral axis and the ventrodorsal axis. But just for simplicity, I will refer to this spatial organization in terms of medial lateral organization. Now, the question we had is, how is the hippocampus organized based on connectivity features? And to address this question, we use meta-analytic connectivity modeling. So we look at voxel coactivation across tasks, and what we see is really this strong differentiation along the longitudinal axis. So this is a view of the hippocampus from the top. So this is the anterior part of the brain, and this is the posterior part of the brain. And if we divide the hippocampus in 2 clusters, we have this head versus posterior region differentiation. But the most stable differentiation is usually a tripartite differentiation that nicely separates the head, body, and tail. We also look at functional connectivity using resting state, functional connectivity across a large core, and we find relatively similar patterns. So really we had this differentiation again along the longitudinal axis, so that differentiate the head, body, and tail within the hippocampus. However, we can see that this type of organization, this pattern, is really different from what we know from cytoarchitecture. So the typical subfield model that I have illustrated here. So this illustration is based on the subfield that you find in the anatomy toolbox. So this is in the MNI space, and here this is also in the MNI space. So you can see that we have a completely different model of hippocampus brain hippocampal organization here. So the next question that came to our mind is, what about structural covariance? So as I said before, structural covariance on the one hand is assumed to reflect functional coupling between brain regions, but the comorphology between brain regions can also be assumed to be influenced by the microstructural properties of the region. So we try to parcellate the hippocampus using structural covariance. And what we saw was first a very strong differentiation along the longitudinal axis. But as we go to a tripartite subdivision, so a subdivision into 3 clusters, what we see then is not a head, body, and tail differentiation, but a medial lateral differentiation in the posterior part of the hippocampus. And this was really reminiscent of the subiculum versus CA subfield differentiation that we know from cytoarchitecture that I have illustrated here. So it really seems that the structural covariance pattern of within the hippocampus would be, on the one hand, characterized by large-scale functional interaction, but also from local microstructure differentiation. To be sure that our pattern of structural covariance was not driven by the specific sample that we use, because to our knowledge, our structural covariance study in the hippocampus was the first study. We look at different cores. So in total, we have gathered around 2,000 participant data. We look at young subject, we look at middle-aged subject, and we look at elderly subject. And we find a really robust and stable subdivision pattern based on structural covariance across the lifespan. And actually, when we compare our studies with other studies from the literature, so I try here to collect the many different parcellation and also gradient studies that can be found in the literature. I think some of them are from people in the audience here today. So I'm really happy to put all this work together and to show the main conclusion that we can get from all this work. So if we look at functional parcellation of the hippocampus, we really see this longitudinal differentiation that comes as the first dimension, the first organizational dimension within the hippocampal formation. So we see it here within the hippocampal formation, but also in a Chase study when they focus on the subiculum. We see it across different studies, and we see it when merging coactivation and resting state in our study. And this is really congruent with gradient studies in which the first gradients usually runs along the longitudinal dimension. However, there is a second dimension of organization along the medial lateral axis that we can see when we go further in parcellation. So when we keep on subdividing the hippocampus, and that can be seen as a second gradient in gradient studies. So this dimension run more along the medial lateral axis. Now, if we look at structural covariance, as I say, we see a slightly different pattern. We still have the main first dimension being the longitudinal, along the longitudinal axis. But then we also have this medial lateral differentiation that is also strongly present in the data. It was not only in our study, but also in another completely independent study that also used different cohorts. And when we look at the gradient of structural covariance within the hippocampus, we also find that the first gradient was along the longitudinal dimension, while the second gradient seems to run more in the medial lateral dimension that we can see here, and that again reminds us of the subfield organization. Now, one concern that we usually have is that could it be that this longitudinal dimension, organizational dimension that we see in functional connectivity is actually not really related to connectivity, so the neurobiological feature that we aim to probe, but that it could be that it is more related to the method that we use, so more related to the use of MRI data or the use of clustering factorization approach. Because the subfield model that we have from cytoarchitecture is usually derived using a boundary mapping approach, which is the typical approach in cytoarchitecture. But in brain parcellation, we use factorization or clustering approach. However, some studies have used clustering and factorization approach applied to MRI data, but not functional connectivity or connectivity data, but local microstructural features. So for example, T1, T2 ratio. And what we see then, when these approaches are used on such estimate of local microstructure, we really see this medial lateral differentiation that again is reminiscent of the subfield organization. And we can also see it in a gradient study applied to microstructural features. And finally, when clustering approach were applied to histological data from the big brain, we really see this medial lateral differentiation again that is really close to the subfield differentiation. And when gradient was also applied to the big brain data, we again see this medial lateral differentiation, but we also see to some extent as a second organizational dimension, a longitudinal differentiation. So this is really interesting that when we have whole brain data in histological work, we do not see only the medial lateral organization, the subfield organization, but also this longitudinal differentiation. So in summary, when we look at hippocampal organization with data-driven approach and factorization clustering approach, we see both dimensional dimension of organization. We see the organization along the longitudinal axis that is really prominent in features probing functional integration of the hippocampus, but we also see a medial lateral differentiation, but that would be more prominent in when using features that probe microstructural properties of the hippocampus. So in sum, by using MRI-based parcellation and gradient mapping of the hippocampal formation that are data-driven techniques. So we don't start with any a priori model. We don't impose a model on our data. This is fully data-driven. We can capture many different dimensions of hippocampal organization, and these techniques provide readily usable outcomes. So the work that I presented today, our work at least, is freely available in our Anima platform. So you can download the maps. For example, if you want to study resting state functional connectivity and have a tripartite subdivision of the hippocampus between the head, body, and tail, you can download our maps in MNI space. This is MNI 152 space on Anima. So with that, I would like to thank all my collaborators for this work, in particular Anna Plachty, who has done a lot of work on the hippocampus during her PhD, and also my collaborator, in particular Catherine Ammons in the Vogue Institute, and the group of Boris Bernard in McGill University. And thank you very much all for your attention. I hope I did not run out of time. I don't think so.
Nope, you were perfect on time.
Cool. Thank you.
All right, so I will open it up. If you want to type your questions in the chat box and have me read them, feel free to do that. Or if you'd like to use the raise hand feature, just so that it doesn't become chaotic and you'd like to unmute and chat, either one is fine with me. And thank you, Sarah, for that talk. It was so cool to see that with a data-driven approach, we can still have these results that are so beautiful and so useful, I think, moving forward as we all try to have innovative approaches to asking questions about how the hippocampus is interplaying with other areas of the brain and how that relates to cognition.
Thank you.
All right, so Rosanna has a question in the chat. She says, what a wonderful overview. I was curious if you had thoughts about the role of spatial resolution on the tripartite anterior to posterior versus subfield medial to lateral approach.
Yeah, that's a really, really good question, because the MRI data that we have usually lower resolution. Yeah, exactly. Functional is usually lower resolution, but the work that I presented has been run in HCP, which already had better resolution than usual studies for fMRI data. And as I showed today, when actually the same parcellation approach is run on, so it was the paddle study published in Neuroimage. So that's, wait, that's that one. So here it was also used, the factorization approach was used also on MRI data, but not on functional data on T1, T2 ratio. So this, we could think that the resolution is slightly better, but it's still MRI. So we are still not at the level of histological work. And still we see this differentiation that really looks like the subfield differentiation in my view. So I think the question may remain open, but there are still a lot of argument for believing that the differentiation, the tripartite differentiation, is not only a matter of spatial resolution. It's difficult to fully prove that based on the data that we have, but at least we have a lot of evidence that would argue that it's not only due to the lower spatial resolution of functional connectivity of functional data. And also the longitudinal organization can also be seen in intracranial recordings. So this is also an evidence in previous reviews that this longitudinal organization is not an artifact of functional MRI. I don't hear you, Kelsey. I think you're
sorry,
muted.
I am muted. I was saying thank you. I was reading Rosanna's thank you, and I was saying, Benno, I see your hand raised if you want to go ahead and unmute.
Yeah. So thanks very much, Sarah, for a very interesting talk. And I would like to follow up a little bit on your response to the first question, because it's really striking that you see both the longitudinal differentiation as well as what you call the medial lateral differentiation. And you said that difference, it's clear, but it may not have purely to do with the resolution. But could it still be that it has some methodological background, that in most of the human studies, the AP differentiation is so clear? Whereas in a lot of animal studies, people tend to focus on the medial lateral resolution because it's so much easier excuse me, with the technology that is used in animals to do that. And the AP axis in animals is the problem because people generally don't record from the whole length of the hippocampus. So if the sensitivity of MRI data and MRI analysis would increase, would you not expect that eventually we're going to see a perfect merger of the two gradients, that we actually have to understand or consider the hippocampus as this very complex structure that actually functions based on these two gradients, that both have important contributions to how we should think about the hippocampus as a functional entity in the brain?
Yes, I definitely agree with your point. We will certainly see both model or organizational patterns superimpose, I think, if we would have higher resolution in MRI. And in particular, I think that the longitudinal differentiation, we would see it when we look at long distance connection, while we could see the subfield differentiation when we look at local connectivity, so connectivity within the medial temporal lobe, I think.
Yeah, I agree. So thanks very much.
Thank you very much for your comment.
All right, well, thank you so much. And I'm going to have Robin now switch over. If we have time at the end, we can open it back up for questions for Sarah. But for right now, Robin, the floor is yours again to introduce Ali.
All right, so I'm extremely happy to introduce our last speaker for today, who is Dr. Ali Khan. So Dr. Khan is an assistant professor of medical biophysics at Western University, scientist in the Imaging Research Laboratories and Center for Functional and Metabolic Mapping at Robarts Research Institute and Canada Research Chair in Computational Neuroimaging. He completed his PhD in engineering science at Simon Fraser University in 2011 and was a postdoctoral fellow at Robarts Research Institute from 2011 to 2014. He leads the CAN Computational Imaging Lab at Western University, focused on the development of novel image processing and analytics using quantitative magnetic resonance imaging. And today we'll be sharing his work, automated computational unfolding of the hippocampus with HippUnfold. So, Ali,
great. Thanks for that introduction, Robin. I'm just going to share my screen now. There we go. I think you should see it now. Get rid of my chat window here. Okay, great. I just want to thank the organizers for inviting me to this really exciting session, and thanks, Sarah, for giving really excellent talk to kick this off. I'm going to be talking about some work that we've been doing, developing more computational methods for performing subfield segmentation, but also more generally giving us another way to examine hippocampal anatomy. Just really briefly, I mean, the kind of work that we do in our lab, it's really about developing tools to quantify structure in the brain. We sort of work at a couple of levels. We work sort of across voxels, looking at morphometry and tractography, other techniques like that, and also zooming into the voxel using different techniques for quantitative mapping, diffusion MRI. And also, quite importantly, we need modalities to sort of bridge these gaps and also to validate what we're doing. And so we make use of histology and other sort of complementary technologies to do this. And so we've been quite interested in the hippocampus subfield segmentation problem because it's also one that really does require a sort of merging of micro scale and macro scale knowledge. So as Sarah really nicely pointed out, these subfields are really the sort of psychoarchitectonic subdivisions of the hippocampus that run in this transverse direction of the hippocampus. And so when we want to examine these in vivo, for example, with MRI, we don't typically have the contrast to really see where these cytoarchitectonic boundaries are. So we really have to rely on indirect rules to do this. And so a good way to sort of see how these rules work is really to look at how they're conventionally applied in the hippocampus. And the conventional technique is to really sort of slice the hippocampus in a coronal plane. And when you do this in the body of the hippocampus, so what I'm showing here are some images from the big brain 3D histology data set that we've segmented recently. And what you can see here is you see this sort of prototypical pattern of progression from the dentate gyrus and the CA subfields into the subiculum. And this is the topological order that we expect to see subfields. And given this, we can then infer based on some geometric properties of where these subfields occur, how to label these different subfields. But this becomes quite challenging when you look at the head or the tail, for example. So in the head of the hippocampus up here, you can see the sort of the classic C shape is no longer visible, or it's at least present with a number of other features. So if you trace out where these subfields are progressing, you can see there are apparent tears in topology. You see sort of the dentate gyrus splitting in a few different places, and the order of subfields is quite different in each case. And the same thing happens in the tail. You sort of you're seeing these apparent breaks in topology. But when you actually relate these slices back to where they're actually being taken in a 3D view of the hippocampus, you can see that these really are just apparent breaks in topology, and they're not. There's no actual break in topology in the hippocampus in 3D. It's just that we're slicing it in a manner that is making it appear as if it were much more complex than it actually is. And so we know quite well from very detailed anatomical studies that the hippocampus is really, it's a really topologically a flat piece of tissue that has undergone folding through development. And and that folding is really occurring on a couple of different levels. There's folding in this sort of transverse direction, or what we're calling proximal and distal to the subiculum, and from the subiculum to the dentate gyrus. And that gives you that sort of characteristic C shape that we see in a slice of the body. But then you also have in this longitudinal axis, you're seeing folding occurring there as well, sort of anterior to posterior. And this even sort of becomes even more complicated. You sort of end up with this kind of cowlick in the head of the hippocampus, or in the vertical component of the uncus here. And to make things worse, you're also seeing a number of these sort of bumps or ridges, and these are really just additional gyrification that occurs sort of just perpendicular to this longitudinal axis. So we began sort of, when we started looking at higher resolution scans with 7 Tesla and 3D histology, we really began to appreciate that these features could be resolved for the most part. And if we could perform a detailed segmentation of the tissue that sort of respects the curvature of the hippocampus, not drawing any subfields at this point yet, but just respecting the curvature of the hippocampus, then we can start to sort of infer where these different axes actually lie. So this led us to some work where a few years back, where we developed this approach to effectively unfold the hippocampus by defining this hippocampal coordinate system. And we do this basically, as I said, so by performing a quite detailed segmentation here in the red here of the gray matter of the hippocampus. And along with these sort of anchor points, so these source and sink regions, which then allow us to, basically index along smoothly along this gray matter tissue to define these longitudinal axes, the transverse axis, along with, although it's quite thin in most scans, but there's this laminar dimension as well. And so, and the way that one way we can generate, this sort of interpolate smoothly between these anchor points is to use a technique called Laplace's equation. So this is effectively just finding a smooth mapping between two surfaces in 3 dimensions. And so this gives us basically a way to sort of simplify the representation of the entire 3D shape of the hippocampus into these 3 perpendicular orthogonal directions. So we have our longitudinal, transverse, and laminar. And this gives us essentially a 3D flattened representation. And if you imagine putting a plane along one of, say, that mid laminar thickness, you end up with a surface mesh effectively. And so, I mean, surface-based approaches have really been used for quite some time now in the cerebral cortex. So back in 1998, David Van Essen had this paper looking at how what the benefits are of doing this kind of surface-based mapping in the cortex. And it really allows you to deal with what are quite individualized patterns of folding in the cortex. And it gives you also a way to better perform techniques like smoothing by treating voxels or vertices that are nearby on the cortex with their actual distance along the cortex. So it's a technique that really provides quite a topologically consistent way to visualize and label brain areas. It also allows you to do things a bit differently in terms of how we measure volume and atrophy. So now we can, instead of measuring volume, we can now look at decomposing that into thickness and surface area. And so we recently proposed this kind of an approach can really be applied to the hippocampus as well if you have high enough quality data and segmentations, and the benefits that we see in terms of being able to deal with the individualized shapes of the hippocampus and their folding patterns can really be realized. And so this is just giving a demonstration of what sort of variation we do see across individuals. So these are actually from data sets acquired at UPenn that were made publicly available. But we can see just this pattern of digitations where you have many or few digitations is one sort of dimension of variability. Also the amount that the hippocampus is curved longitudinally, or how much sort of tissue you have sort of anterior versus posteriorly can really change this global shape. And if you're not dealing with the shape in 3D, it can make subfield segmentation especially challenging. So one piece of evidence that we found really compelling for this unfolding approach was when we took data, sort of labeled data from our data sets, from our 70 data sets, where we had subfield labels and a number of different subjects labeled, and we mapped them to this unfolded space using just each subject's intrinsic mapping to these three coordinates. We ended up seeing there was quite a lot of just intrinsic alignment that we get, both in the subfield boundaries. So here, what I'm showing in this dark gray is sort of just how much this boundary is varying across our subjects in this data set, along with just how microstructural metrics appear once you bring them into this space. So seeing these metrics that line up with subfield boundaries and that also have good alignment to produce these sort of average maps was evidence that this technique is really doing a good job at intrinsically aligning anatomy. So in the last few years, we really put a lot of effort into trying to make this technique more widely accessible and usable on data sets without requiring the detailed kind of manual segmentation that we had performed in our initial work. And to basically use this approach not only to perform subfield segmentation and provide volumetric labels of subfields, but also to really take advantage of everything you can do with surfaces once you have surfaces of the hippocampus. So I'm going to talk a little bit more about all the different aspects of this work and how we actually perform this unfolding. So the main way that we automate the approach is really to use a deep learning segmentation to perform this initial tissue segmentation. So we make use of the NoNu UNet platform for helping sort of automatically infer an optimal unit architecture. And this has been shown to work really well on a number of different domains and data sets. But it allowed us to basically train a number of different networks on different types of images. So using the Human Connectome Project, T1 and T2, and even diffusion weighted imaging data, we were able to, given a fairly large data set of labeled segmentations, and here we're not labeling the subfields, we're just labeling this essentially the tissue of the hippocampus, which includes the SRLM, which is quite critical in our approach because it allows us to really follow the curves of the hippocampus, along with these endpoints, which we use for defining our Laplace coordinates. So with a number of these segmentations to train on, And these segmentations were sort of iteratively developed through a series of initial manual segmentations followed by corrections from an automated approach, because it's quite a lot of subjects to have to segment. And this is work done by Jordan de Kraker during his PhD with Stefan Kohler and myself. And so we're able to really quite robustly produce these initial images that we could then use to produce our coordinates. And so the coordinates themselves are produced with the Laplace's equation, as I mentioned, but these then allow us to effectively find a mapping or a transformation to go from a native hippocampus to this unfolded hippocampus. And then we also invert this to be able to go the other way around as well. So we have a way of going between this, for example, here I'm showing this coronal slice of a hippocampus. And when we transform this into our unfolded space, we end up with something like this. So here, this is just the same subject actually. So the intensities you're seeing here are really just effectively sampled at the mid thickness of this hippocampus. So you can see at this darker region here, this is the subiculum, and you can see the subiculum is darker here. And of course, we have a way to go back and forth. And so how does this actually, how do we use this for actually performing subfield labeling? And how does it compare to conventional approaches of doing this? So in a conventional subfield segmentation, that's done in the native space, you're trying to infer some function effectively to perform a labeling of this image. So this function of the image of X, Y, Z coordinates in this image to give us our subfield labels. And this in general it's a tricky function to define. I mean, if you're doing it with manual labeling, as I described before, the rules that you might have to impose in the head and tail can get really complicated. And there's a lot of intersubject variability in those areas as well. If you're doing this with automated labeling, so that often involves a number of nonlinear atlas registration steps or using a label training set. You could use a UNet like we do in this case to actually learn how to label subfields directly, but then you would have to label a large data set with subfield labels. And if you were to change your protocol later on, you would have to retrain and label again. So by labeling in the unfolded space, so this labeling function then just becomes a simple mapping. So instead of a complicated function that involves a good deal of nonlinear registration and label data sets, you can really just use essentially a lookup table to define where these subfields exist. So it's worth taking a closer look at how this actually relates to existing manual protocols, especially given that this is the hippocampal subfields group, and one of the goals of this group is really to, or the primary goal is really to come up with a harmonized subfield segmentation protocol. So I'd like to just dig in a little bit about how this approach is sort of analogous to what others are already doing. And so if you think of sort of taking a sort of vertical line in this unfolded space, that's really like taking a coronal slice in the hippocampus. And when you do this, you can see that the subfields, the subfield boundaries that we get are really just defined by a proportional distance along the hippocampus or along the SLM. And this is actually an approach that has been proposed sort of independently of us as well, as a way of, as a histologically validated segmentation protocol to apply in the body. It was also recently reproduced by Robin here in another 9 specimens. And And so you can think of our unfolding approach of subfield labeling as one that's quite analogous to this, except generalized to 3D. And similar to the Harmonized protocol that's been proposed so far from the hippocampal subfields group, this is one that looks at essentially sort of defining a crosshair and angles relative to this. You could think of, if you consider an assumption where the SRLM is sort of circular in shape, then this really has a direct relation to the arc length. So the SLM distance could really be the arc length here. So having this angular representation is quite analogous as well. I do want to also point out how our labels generally compare to what else is out there in the automated world. So ashes and free server, I would say, are the two most popular subfield segmentation methods out there. And of course, with ashes, you have a number of different atlases you can use. So this is just one of them. But the one benefit that we have with HippUnfold is it's much easier to label in the head and the tail. So a lot of these softwares really don't even attempt to label in the tail because of the challenges here. But here we can provide a segmentation that is sort of internally consistent. And I think what makes it even more sort of really relevant to this community is that the subfield boundary definitions that we use are really quite customizable. So we've been making use, our sort of in-house gold standard or silver standard to some is this single subject big brain 3D histology data set. So this is one where we can see the cytoarchitecture, so we have a good idea of the boundaries, but it is just a single subject. So it certainly has limitations, and there could be distortions involved as well when doing the 3D histology reconstruction. So we do have ways that we can create new atlases from basically any kind of segmentation just by unfolding those segmentations. So we've done this just very recently to use an existing atlas, the atlas that was used in ashes for 35 subjects using 7T data. And just by unfolding that data, we can produce a probabilistic atlas, which can then be used in our software as well. And this is just showing what that maximum probability labeling of that atlas would look like. I just want to make sure I'm doing okay for time. I think I'm doing okay. I'll just have a couple more minutes here. We also wanted to see how well it is generalized to other data, and it does. It does surprisingly well, even with T1 weighted images. So we have models that were trained specifically for T2, and we also have models that were trained for T1. You can see some more details in our bioarchive, but we find that actually our results on T1 weighted data sets seem to be actually even more robust. The failure rates are lower. It also does work well when you have a more conventional hippocampal protocol, one with, for example, thick slices and high in plane resolution. And we end up upsampling the data for internally to make that work a bit better. And just one final point about the methods. So one important aspect of this is we also enable the use of surfaces. So by defining a tessellated mesh in this unfolded space, we're able to get a corresponding set of vertices in every subject. And we have this for a number of different densities, so that this is a way you, once we have imaging data that's high enough quality, you can really map anything you want to these surfaces. So you can map functional data or diffusion data. We also generate, based on the surfaces we have, we can generate thickness maps, curvature maps, and gyrification maps. So these provide, new metrics that we can use to really explore structure and function. So I did have a few slides for just sort of what we're doing with our techniques, but I'm just going to skip those and maybe just basically flip through these sort of like 5 seconds at a time just to get a teaser. These are all going to be presented at OHBM this summer, so hopefully you can come find us and talk to us about the hippocampus. And if you're looking for the code itself, we have it available online. We're also happy to help people use the software. So give it a try. Post an issue if you have any trouble with it, and we're happy to help. So with that, I will end.
Awesome. Thank you. That was so cool to see you go through all the different ways in which you can take, again, a data-driven approach with some complementary aspects of histology and get these really, really interesting and, in some ways comparable definitions. So before we end today, I'm going to allow there to be time for 1 or 2 quick questions at least. And yes, and so Laura Vissa has a question in the chat. I don't know if you've already read it, then.
Yeah, I see that. Yeah, it does work pretty well. We didn't train it with anisotropic data, but so far from what we've seen, it does work well. But happy to test it out some more. For example, it worked on the 35 Magdeburg atlas subjects. It worked fine.
Awesome. She says, thank you. All right. Does anyone else have a quick question before we close? And I will also, when I upload this to YouTube, make sure that I have ways that you can reach out and contact, like all those links that were present on Aldi's last slide. All right, Sarah.
Yes, thank you very much for the very nice talk. So this is really impressive, the fine segmentation that you can achieve. I had one question that you partly addressed. I was wondering about the generalizability of the technique. So as far you have mainly used HCP data, right? And we all tend to focus on HCP because it's high quality data, but you said that it could surprisingly well generalize to 70 data, but was it also HCP or was it a completely independent data set, the 70?
Yeah, we've run it on an independent 70 data set, so some data from Magdeburg. We've also, it also generalizes fairly well to like lower quality data. So I don't know if I can recommend people to use 1 millimeter T1s, but it'll produce results on 1 millimeter T1 data. But it's unclear how specific those results will be. But yeah, certainly it does work on 70 data from what we can tell, but we haven't tested hundreds of data sets. Yes, we've tested,
of course,
probably less than 100. Yeah.
Thanks. Thank you very much. If I can, I would also ask another question.
Yeah.
So I'm particularly interested at the moment in understanding hippocampal development. In kids. I was wondering, I really don't know about such technique in kids. Like, do you have any ongoing studies that would provide such segmentation in kids' data?
Yeah, I've got a couple of things I skipped over, but we have had some collaborators. So Conrad Waxdell and Sophie Adler and their student Mathilde Ripart, they've applied the technique to pediatric epilepsy cases and have had good success in being able to segment the controls and also to identify abnormalities in the patients. We've also and we had to sort of generalize in a sort of iterative way, but we have it working on neonatal data as well, and this is going to be presented at OHBM. It still is not as accurate, and it fails more than it does for adult data. But I think it's a promising step. that we can even segment subfields in neonatal data. And this is, of course, very high quality neonatal data. This is, where is it? Yes, this is developing human connectome project data. So, but yeah, that's something that we're interested in as well, just applying it to the broader age range. So we are, we have applied it to sort of data sets from 5 to 100 years old, and we haven't done any strict validation of the results, but the method works, and we are able to see sort of curves that are what we typically expect in things like thickness as well.
That's great. Wonderful. Thank you.
Yes, thank you. And thank you to you as well, Sarah. It was such a, I think, really great session, and I'm glad that we have this to reference back. I'm looking forward to seeing what both of you have coming up. I know that, Ellie, you said one of yours is preprint, so make sure you let us all know when that is officially out. And for those interested, like I said, I'll make sure that any of the links that were in there in the talk, especially on those last few slides, are up when I put this on YouTube by Friday, because if you've been here before, my computer takes forever to upload. But with that, I just want to thank everyone for coming in again. And the next time we will see you is going to be on the 27th of April. And that will be featuring our own Menno Witter, who is in here looking at the entorhinal cortex circuitry in animals. So I'm looking forward to seeing you all then. Until then, have a great rest of your Wednesday and a great rest of the week.
Bye. Thank you.
Thank you very much. Bye-bye.